In 2011, when I was just about a seventh grader in middle school, I was introduced to this little program by my biology teacher. And at first glance, it doesn't look like much. It looks like a little worm. And I remember thinking, hey, this is pretty cool, but why is there a whole website dedicated to this model? And then I found this bar on the side and I started scrubbing through. And I could see that we could get a view into the whole anatomy of this little worm. Its muscles, its reproductive organs, its nervous system, etc. And for a minute, I was really, intrigued. But unfortunately, at the time, I really didn't know what to make of it. I mean, uh, here was a nervous system of some animal I didn't really know too much about, and a bunch of body parts, and seemingly nothing to do with it. And so I went back to, just doing what middle schoolers do. And now, about a decade and a half later, I'm revisiting this creature, C. elegans, this worm, in a whole new light. I'd like to start by going all the way back to Sidney Brenner, the guy who single-handedly, pretty much, brought C. elegans nematodes into the biological bio-limelight due to his fascination with sequencing the C. elegans genome. In 1963, he requested a culture of nematodes, and from then on, basically, C. elegans became a model organism. A model organism for us to poke and pry at, and it definitely did bring with it a number of explosive successes. To begin with, in 1999, Sidney Brenner's dream came to life, and we have the entire C. elegans connectome. So you can find that on this older website, Wormbase, and you can see on their newer website, Alliance of Genome Resources. You can basically search up not just nematodes, I mean many animals for which we have genomes, but nematodes were the first animal to have a fully sequenced genome. And nowadays you can just search this up. For example, I may be interested in all the signaling receptor genes in the C. elegans nervous system. And here you go. You can get 7 pages of it right in front of your face. And from there, research continued to explode. We'll revisit what happened in 1986, which was John White's mapping of the entire connectome, and we'll talk about what that was. And so there was an explosion of research on nematodes in many different directions. We figured out a lot of things about its nervous system, about its other body parts and its cells, and we even began to simulate how it moves. So in 2007, Andrey Palyanov started the Cyber Elegans project. And in 2009, he released this video. What he was able to demonstrate is how to translate the body plan of C. elegans, the nematode, into a collection of muscles and springs that, when activated correctly, could produce a very similar motion to what we saw in the real nematode. From there, this sort of initiative exploded and evolved into cybernetic, which is one of the current iterations that he has of this project. Now everything is simulated with particles. It's incredibly detailed, etc., etc. And now also this is part of a much larger project called Open Worm. And so we'll see that. I'll show you a little bit more eye candy. So this all here is simulated particles making a simulated worm. You can also check out the paper here. But, you know, he didn't do it alone. For some reason, over those few years, a bunch of people, including Sergei Khairulin and Giovanni Adili and Stephen Larson, came together basically to spawn this project called OpenWorm. And that very project is the one that I saw in my viewer in 2011. What I was able to get a view at was a project that spanned much, much larger than just this anatomy viewer. And so if we look at a 2018 review of OpenWorm, you can see how much stuff they've actually been doing. They've tried to both map a lot of the anatomy and structural data, but then translate that into simulation tools, connect that to the simulator that we saw before, connect that to web interfaces, and they've also gone on to try to simulate the real neurons in high fidelity. The biggest problem with OpenWorm is that it's pretty much all over the place. You can see all these different repositories that encapsulate OpenWorm projects, some more recently maintained than others. You can see a variety of their resources here. So if you check out the connectome on Open Source Brain, you can see a bit more of a detailed view of the nervous system on its own and the ability to select individual neurons and, in theory, also simulate the nervous system here. But I can't really, another problem is these tools are extremely unruly to use. At some point, they even had this little guy, which you can see the teaser trailer for here. They sort of dreamt up this idea that you could have, just through a web interface, a complete view of the simulation of this entire nematode right in front of your face. And you should be able to see the muscles and the nervous system and everything sort of co-activating and alive. But unfortunately, it seems like these bits really never came to fruition. And unfortunately, if you look up wormsim.org right now, you will find that there is no website to go to.